So good evening, everyone. Myself, Rishampreet Kaur, and I'm a student in BTech Biotechnology, first year student. So first of all, I'd like to thank all the organizers and the moderators of this TBS BioCon event for, providing such an important platform for, budding people who have an interest in science like myself. Also to present our views and have discussion and forums like this. It's a great platform. Thank you so much. During the time allocated to me, we shall be talking about cloning. So, as the name suggests, cloning, by define, if we have to define, cloning in biology is a process of producing similar populations of genetically identical individuals that occurs in nature. Yeah, it occurs in nature. It might be quite surprising for some that cloning it's occurring in nature, although it might seem to be a quite kind of a technical or a scientific term, which we might think it happens only in the labs, but that's not the case. Cloning, it occurs in nature when it occurs for the organisms like bacteria, insects, or plants when they reproduce asexually. For the bacteria, it's a very common example. We know in the case of like fission, especially in bacteria or the other modes, like whenever two daughter cells are produced, the two daughter cells are the exact copies of their parents. So in that case, whenever something is genetically exact copy, we would call it to be a clone. So the daughter cells could be called the clones of the parent cells. That would be an example of naturally occurring cloning in the microorganisms. For the case of human beings also, we have certain natural examples of cloning, which is the case of identical twins. It is for the fact because they have the same genetic composition. The identical twins, they like the same zygote. During the stage, it is split into half, leading to the formation of the two different individuals that are genetically identical. So next up, we would consider the history of cloning. Like today, we have great, developments and experiments in the field of cloning. But back some years back, the idea of cloning was absolutely unknown to the scientists, and it was only in the year 1953 when the structure of DNA was discovered to very famous scientists, James Watson and Francis Crick. They gave the double helix model of the DNA, and it was after the years after them that many researches were carried out on the basis of the DNA, genes, and the genomes. And after the structure of DNA was discovered in 53, in 1993, human embryos were first cloned. That was first artificial embryo being cloned, and on July 1996, that could be called a breakthrough in the events of cloning. Dolly was born. Dolly was basically a sheep, which is the first mammal ever to be cloned. And she was born on 5th July 1996, which is almost 25 years from now. And we shall be discussing about it later in the further slides, like all the mechanisms involved in the production of Dolly. So next up, we have certain different types of cloning. We have DNA cloning, reproductive cloning, and therapeutic cloning. Also, we need to study here the mechanism of cloning, how the cloning is actually taking place. That we shall be covering with the types of the cloning, like which type of cloning, how that is taking place. So first up, we have with us DNA cloning. DNA cloning, just from the name we can guess, it is a process of producing multiple copies or the clones of DNA, or might be a small strand of the DNA, which we call in simple terms a gene. So that would be DNA cloning. In the method of DNA cloning, we use a vector. So vector would be something that would be used to transfer the part of DNA that is to be cloned into some host cell, which would produce multiple copies that would regenerate number of recombinant DNAs. So the vector, most commonly used vector is plasmid. Plasmid, as we all must know, it is an extra chromosomal DNA which is present inside the bacterial cell out of the nucleus, and it is a self-replicating, autonomously replicating piece of a piece of DNA. So we use plasmid, and plasmid is circular and round, and we use certain endonucleases, restriction endonucleases, that is the enzymes. We use those enzymes to put cuts on the plasmid, and then the emptied part of then after the plasmid is cleaved, we also generate out a DNA that we need to copy, that we need to clone from the eukaryotic chromosome. In that eukaryotic chromosome, it is again done with the help of endonucleases. That cut part of the DNA is then ligated into the plasmid with the help of another enzyme, that is the DNA ligase. It is an enzyme which helps in the ligation, that is the joining of the two DNAs. So now we have with us a plasmid that has in it a small piece of the DNA that we want to clone. Further, there are many mechanisms, like whenever we recombine the DNA, this we would call recombinant vector DNA. When we recombine the DNA, during some time it gets recombined. Sometimes some DNAs, they are not recombined. So that's a different topic. Those can be separated with the help of blue white selection. We use X-gal as medium, and then the colonies are grown on them. So the colonies that produce white color, because they were not able to produce any product, so those mark the recombinant DNAs, and the other one that produce blue colonies, because they were able to form the product, that is the beta galactosidase. Those form the blue colony. So that's a different topic. So now we have with us the recombinant DNAs. Now those recombinant vector DNAs, they are introduced into the host cells, and the host cells normally used is the E. coli bacteria. E. coli anyway is the most widely studied bacteria, and in the E. coli, its replication time 20 minutes. So it replicates to produce multiple copies, and we get with us multiple recombinant DNAs, and from that we can cut out the pieces of DNA, and we get various clones of DNA, and this we would call DNA cloning. So this was the first type of cloning. As we discussed earlier, we have three different types of clonings. The second type of cloning that we have with us is the reproductive cloning. Reproductive cloning is, in a way, one of the most controversial type of cloning, because reproductive cloning means it is a technology that would regenerate an entire organism, not just a piece of DNA, not just a gene, but it would clone the entire organism. The example that I've taken here is of Dolly sheep. Dolly sheep was cloned using the method of reproductive cloning only. So we can discuss the example of DNA Dolly sheep only to discuss how the method of reproductive cloning is carried out. For cloning Dolly sheep, basically one sheep was taken and its normal somatic cell. The cell was taken from the mammary gland, which was a normal somatic cell. So that cell was taken, and from that cell, the donor cell that was taken, and then we had another oocyte, that is the egg cell, from another donor, and from that egg cell or the oocyte, the egg cell was taken out. That was enucleated egg cell. It did not have the nucleus. And from the somatic cell that came from the mammary gland, we took only the nucleus. And after that, with the help of techniques, we have different techniques we can use. One is like electroporation. We have, with the help of electricity, we are able to create a kind of pores in the membrane, and then it can be fused there. Also, we have certain other methods like micro injection, micro pipette, and micro needles. Using all those sophisticated instruments, we were able to to take out the nucleus from the somatic cell of the mammary gland, and that nucleus was then fused into the enucleated egg cell, and then we had an egg cell, and then further the process of fertilization was carried out, and then led by the production of the embryo, the zygote, and then a cell division, and then an embryo was formed. Until then, the process was being carried out in the laboratory. After we have the embryo with us, the embryo is then implanted into a surrogate mother, which was a sheep, and that sheep then gave birth to Dolly on 5th July 1996. And it wasn't really an easy process, because it took the scientists almost like 276 to 277 attempts to produce the clone of Dolly sheep. And during its life, Dolly was, as expected, just like any other normal sheep, it lived its life. And during its lifetime, it produced 6 lambs, and among them was a pair of twins and a pair of triplets. And Dolly, she was shot down at the age of 6 because of its incurable pulmonary infection. Although almost every the scientists believe that this has nothing to do really with the fact that it was cloned, because at the end Dolly died of an infection, that was a lung infection, the pulmonary infection, and it is quite common in the livestock that are being used for experiment in the labs, and they are not exposed to the natural environment. And the third type of cloning that we have with us is the therapeutic cloning. This is by far the most useful type of cloning if put to use. And for studying therapeutic cloning, we need to study an important term, that is the stem cells. The stem cells, or the cells that are present in the embryo, we call them the embryonic stem cells. So what are they used for? A very common example, all of us, we have, we arise from a single cell, zygote. So zygote, it is initially one cell, and then it divides, then we get certain stages like morula, blastula. And the scientists believe that up till we have a stage of 100 cells, we have certain stem cells, and those stem cells, they have the ability to divide and then differentiate. There is a difference between division and differentiation. Differentiation would mean that one cell is capable of giving rise to various different types of cells. Like, one, we have a group of stem cells, they are differentiating. Some are forming our limbs, some are forming our liver cells, some are forming our lung cells, some are forming our nerve cells, the neurons, and various other types of cells. So the stem cells have this capability, whereas other cells of our body that are differentiated cells, they don't have this capability. Although the liver cells can regenerate, if the liver cell was to divide, it would divide into to form other liver cells only. So they don't have the ability to differentiate further, as they are already differentiated. So the scientists then, they have been trying rigorously in the process of therapeutic cloning, for a patient whose any organ has been damaged. They have been trying to, with the help of the stem cells, embryonic stem cells, they are trying to use an enucleated donor oocyte, as was taken in the previous case, and then a somatic cell from the patient, and to produce the embryonic stem cells of that patient. So we have this of that patient, and now they can be differentiated to produce whatever organ that was desired inside that patient. So a healthy copy would be produced of that sick person's tissue or the organ for transplant, and then it would be a better alternative for the organ transplantation, because like we have seen various different cases of graft rejection in the organ transplantation. So this was about the therapeutic cloning. That is the third type of cloning. And next up, we have with us certain applications of cloning. And first of all would be the production of recombinant proteins. And we can produce various kinds of recombinant proteins with cloning. We are able to produce one protein, and then we can create multiple copies of that. Then we can create a number of transgenic organisms, that is GMOs, the genetically modified organisms, and then we have been able to create many of them. For example, we have created some herbicide resistant crop plants, and then we have a glowfish, that is a very common fish in our homes. It's like just in the aquariums we can have. So it is a GMO, genetically modified organism. And next we have gene therapy. Gene therapy is another very important application of cloning, or RDT in general. And gene therapy, the first gene therapy was given to a 5-year-old girl who had ADA, and then her gene was changed, although it was an incurable disease. But if the cloning, with the help of sophisticated techniques, it is being done at the embryonic stage, the patient could be recovered from it permanently. And next up, we have a question that, do cloned animals always look identical? Although our answer to the question might be yes, they should look identical because they have the same genetic makeup, but that's not really the case. Genetic animals do not, or the clones do not always look identical. A classic example would be the first cat, that was CC, the first cloned cat, CC, the female calico cat. It did not look like its mother. Here we have picture also. So the reason for this is, one reason is that the environment, it plays a big role in how the organism turns out. Not only the genetic material like genotype and phenotype both have got a role to play. And secondly, also in the case of this cat particularly, um, the genes, they do not have a major role to play in the color of the cat. That is something present on the X chromosome. That's a different topic. So it is, we cannot necessarily say that all the clones would look identical. And next up, we have certain potential applications of the cloned animals. And potential applications would be, first of all, the major application that we have been experiencing and using it a lot many times would be testing new drugs and the treatment strategies. they have the same genetic makeup, so the responses should be uniform. Suppose we are to test a particular medicine or a treatment, if we are able to if we are to test them on humans or some different other naturally occurring organisms, then the responses would be different, because all of them would be having their different physiology and their different genetic makeup, so the responses would come out to be different. But for the clones, since they all have the same genetic makeup, we would expect the responses to be uniform, and the experiment or the research could be carried out more easily. And secondly, scientists, they have been trying, although I don't think they have been successfully able to do so, but they have been trying to build populations of the endangered or the possibly extinct species. There have been a news some years back, the scientists, they said that they will be able to, clone the extinct copies of species of rhinoceros, that they would do that. And in 2001, the researchers, they produced first clone of an endangered Asian ox. But the Asian ox that was produced, its lifetime wasn't much. I think after 3 weeks it died. And also, it's not just that it's always good to clone animals. There are certain difficulties also, because the clones, they lack the genetic variability. And about this we shall be discussing later on, in maybe in the next slide. So next up we have with us the drawbacks of cloning animals. It's not just that cloning animals only has its benefits, we also have certain drawbacks of cloning animals. So drawback of cloning is very inefficient technique. We can take this from the example of Dolly sheep. Like 276 clones did not turn out to be successful. So it is a very inefficient technique. The efficiency is very low. And secondly, it might have chromosomes that are shorter than usual. So we know for a normal life cycle, for a chromosome at the end, we have a telomere string, and this is of the chromosome, ultimately it begins to shorten, and then which brings basically the life of a person to an end. So, but in the case of cloning, the chromosomes, they are shorter than usual. We can take the example of Dolly sheep. Dolly sheep, it was like the normal lifespan of a sheep would be 12 years. But for the case of Dolly sheep, it was just 6 years. The reason believed to be that, although she died afterwards due to a disease, but its chromosomes were shorter than the usual chromosome a normal sheep would have. And third up, we have another problem with the cloning, that is, it would increase the birth size and defects in vital organs like liver, brain, and heart of the cloned organisms. So, till now, we have been talking about cloning animals. So, we have another interesting topic, that is human cloning. So just imagine 3 or 4 copies of yourself. I have another myself, going to the college, and another copy of mine who's playing in the playground. So how would that be? Or take another vague example. In the future, we have a couple going to the hospital and, telling them all the specifications that in our kid we would like to have, say, blue eyes and blonde hair and gene of intelligence, as they say, especially for Indian parents, and all these things they would want to have in their kid, and they are going and telling the doctor or the geneticist, and then they are having designer babies. So although this idea may seem appealing to some, but for others, yet many others, it is a great ethical concern. So coming back to human cloning is basically the creation of a genetically identical copy of a human. This term is generally used to refer artificial humans. So it is the production of human cells and tissues. So it's not just that it has ethical concerns, it has some pros also, human cloning. First of all would be solution to infertility for the couples that cannot, those who cannot have, babies naturally, or for the same sex couples, it would be especially not generally prefer to go for it. This cloning could be a solution to infertility. And second, we have organ replacement, as we have already discussed in the case of cloning before, that we clone an organ with the help of stem cells, and that clone could be used. And then we have genetic research. Say for genetic research, like we can do and research if we have same clones that have same features, so same possible outcomes would be expected. And next up, we have selective human traits, although it is quite controversial whether to put this in pros or in cons. But anyways, there could be some, disastrous human traits that that families, or there could be some diseases that the families would want to be removed in their bloodline. So that could be done with the help of technology now. And not just the pros, it has cons also. So we have reproductive cloning, the designer babies, as we discussed earlier. So according to many writers and scientists, the idea of clones is like having clones is like having brainless monsters. So reproductive cloning, designer babies. And next up, we have another major problem, that is the unique identities. So since the clones, they would have the same genotypic and to some extent phenotypic features, so they would not have any unique identities. This would pave a very big problem in both ethical, and also it would increase crimes, like their identity wouldn't be identified. And next up, it would weaken the diversity and ability of adaptation. According to any ecologist would say, as all of us also know, we believe that it is only due to diversity and adaptation that we all are here, and we are because we are what we are. And the world started as a single celled organism. I mean, everything in the world started as a single celled, and then it further evolved and evolved. And it was due to the diversity and the adaptations, basically, that everything diversified into all the organisms that we have today. So if there would have been clones of everything, then we wouldn't have this diversity. So it would weaken diversity and the organisms would not adapt. And the last major con, there is yet another concern. Like you can imagine, just imagine people like Adolf Hitler, they are coming in groups, that is the clones, and then they are invading the world. So that would be a major con of the same. And the last con that I have written is humans acting as God. Although many people in science do not believe in the idea of the God, but that anyhow, so we have already tried to do many manipulations in all the naturally occurring things. Like we destroyed the forest, and then we have seen many disastrous effects of the same. And also the coronavirus, which brought the world to a halt for almost more than a year now. So although it was at some point said that coronavirus is a virus that had been produced in the lab, although it wasn't produced, they weren't able to prove that later on. But anyways, it could be used. Any virus could be, now we know it can be used as a bioweapon and threaten the entire world. So that is kind of manipulations with the natural things. So that is humans acting as God, the covering the act of God. So that is not really a good thing. So that would be a con of human cloning. So the question to answer whether human cloning is good or bad, I would leave it to all of you. Whether what do you think, it's a good or a bad thing? So I think that's all for my today's presentation. Thank you so much. And this is my contact. You might contact me here. Thank you. Any questions?
Yeah, there are a few in the chat box. The first
Uh,
one, yeah. What are the possible errors which cause failure of cloning?
Possible errors which cause failure of cloning. Possible errors could be at many stages. Like we discussed, the plasmid. It was, a DNA was being introduced into the plasmid. So error could be at that stage also. Like we have to very carefully use all the equipment, like the endonucleases, the restriction enzymes, the ligation, and then choose only the recombinant DNAs and then use it to be. So that could be one of the possible errors. And after that also, like we have, it has to be a very sophisticated process of the embryo transfer, that is to be transferred into the surrogate mother. And then we also got to fuse the enucleated cell and the nucleus, so that requires all the very sophisticated instruments. So many errors are possible in all in any of the time there.
Thank
I
you.
think, yeah, thank you, ma'am.
Oh, no way. Yeah, one more question. For drug testing on cloned animals, is it possible that the external conditions affect the testing results? Have we ever seen drug testing results in cloned animals?
Uh, the question is, for drug testing in cloned animals, is it possible that external conditions affect the testing results? Well, ma'am, I would say I'm not really quite sure of the answer, but I think the external conditions would affect the testing results because, as I discussed in one of my slides, the color it came out to be different because the environmental conditions they do make a difference. So on the cloned organisms, I think the external conditions they would affect the results. And for the second part, have we ever seen different drug testing results in cloned animals? I would say that I haven't read about it. I'll have to read on that, and then only I can answer it because I would not really say yes or no to that. But for the first part, I think that external conditions would make a difference because that did in the case of at least the first clone cat, the species that we talked about.
And are there any drug testing results in cloned animals? Was that a part of the question or a separate question?
Uh,
I'm not sure.
Actually, this is a separate question. The example that
Okay.
I took was a simple cloning, and this one is about the drug testing. So I think that's a different thing.
Mm. Yeah.
So Ranjana has raised her hand, so I think she has a question.
Yes, mine is more. I'm just. It's out of curiosity. So it was a very nice talk, but in all your examples, you showed that the cloned product was not similar to actually the source of the clone. So is it a valid worry to have? I mean, is cloning really, has it really stuck on to the to what it is supposed to be? I mean, we are not in all the examples that no duplicates have been created. They were all unique in characteristics. So maybe isn't cloning just another form of reproduction of some sort? I mean, not duplication, but just out of the mm, uh, just
You
another form
said
of
is
reproduction.
another form of.
Reproduction.
Okay, okay,
Just
okay.
I mean, it's not a form of duplication. That's what I was. The whole
Okay.
worry about
So
the whole hype about cloning is.
yes. So I think maybe I missed that point. But the prominent example that I took for the Dolly sheep that I took, it was like all the cells we have been producing from a somatic cell and an egg cell, and all the clones that we have got, they were the exact identical copies of the one of the characteristics of the one that we took from the somatic cell. Same was the case for the sheep, for the example that I took, maybe for the stem cells also. In that case also, the products that we are to get, they will be exact copies of the somatic cell that would be taking, not the nucleus part. The one from which the nucleus would be coming, not the one from which the cytoplasm would be coming. The example that I took would not be the same, was of the first cloned cat. And it was in that case, it wasn't that the cloned animal wasn't same because it didn't have the genetic makeup. It was just that it did not have, because it was affected to some extent by the external environmental factors. And in the case of the cat, it was basically an X chromosome controlling the color of the coat of the cat. So because that's why it produced some different results. As far as I know, all the as far as I have considered, I tried to put up there the other examples of the Dolly sheep, or the other examples that I tried to put up would be basically the clones in this way, that all the same characters would be of those from which the nucleus would be coming. So they would be the same exact identical copies only, until and unless affected by some environmental conditions.
Okay, thank you. This was much clearer. Now I get it. Thank you. Thank you so much.
Thank you. Thank you so much.